So now, turning to Larry, he's a neuroscientist at the University of Southern California who studies how the nervous system controls thinking, feeling, and behavior. That's just about everything, right? And since graduate school, Larry has been interested in clarifying this organization of brain systems that motivate all these intellectual and emotional behaviors. He's an elected member of the National Academy of Sciences, National Academy of Arts and Sciences, American Association for the Advancement of Science, and the Grolier Club. He's served as president, most importantly, of the Cajal Club and of the Society for Neuroscience, and is currently Secretary General for the International Brain Research Organization. Plus, he's a really good guy. So help me welcome Larry Swanson.
Thanks for that great introduction, and actually the chance to come and talk to you tonight, really about my hero, this guy, Santiago Ramon y Cajal. And I'll try and give you an idea of why he's my hero, but part of it goes back to this really weird picture from like about 1992, when my wife and I spent about 10 years translating Cajal's great Bible of neuroscience. It's really probably the starting point, the foundation of the way we think about the brain today. And it was a huge, huge effort. It's about a quarter less than the Bible in terms of the total number of words. It was huge. And believe me, if it wasn't fascinating and really interesting, there's no way anybody could actually do this. So I have spent a large part of my life thinking about this guy, Cajal. And here he is. This is a self-portrait that he took after he won the Nobel Prize, basically. A really stern-looking guy there. Who was he? You know, who was this guy? Well, he's not a household word. He's not like Darwin or Pasteur or Freud, where everybody has heard of them and sort of knows what they do. Cajal is every bit as great, but it's a little harder to understand exactly what he did. When I tell you that he invented the nerve cell, more or less the way we think about it now, doesn't mean a whole lot to you, but it's actually the foundation of actually how we think about the nervous system. And so I'll go through some of his art and life and explain a little bit how he got where he did. But I want to put that actually into a little bit of historical perspective so that you can get maybe a better appreciation of where, you know, the background of what he actually did. And for this, I'm going to start, history, you never know where to start exactly, but I'm going to start with this 28-year-old guy here from Brussels. And this was a portrait from 1543 when he was 28. And he was an anatomist. He wrote the first great book of medicine in the Renaissance. And so it's still considered one of the two best books ever written in medicine. What it was a huge illustrated textbook of human anatomy. It was actually the first one. And you can still use it today, basically, to teach anatomy. And he was an extremely interesting guy. He came from 4 generations of physicians. He was Charles V's personal physician, an incredibly accomplished guy. And what he did, this is a drawing he did of the human nervous system. And he was a pretty good artist himself, but he was in Padua and went over and got artists from Titian's studio to come to the medical school and participate in these dissections and draw exactly what they saw. So these are actually, this book, one reason it's so good is because the art is phenomenal. This is among the best depictions of human body that you'll still find. But the point of showing you this is the nervous system. This is what neuroscientists are trying to explain. How does this work? What you can see up there is the brain up at the top, and the spinal cord comes down the middle there, and then you have the nerves going out to all parts of the body. In fact, when you take away the rest of the body, you can still see the outline of the body there because the nervous system goes and does it all, moves all the muscles, et cetera, et cetera. So as neuroscientists, we go back basically to a drawing like this and say, how does this actually work? You know, what are the parts? How do those parts work? How are they interconnected? It's a system. So as a biologist, you actually want to say, well, how does it work? And we know how the heart works, we know how the stomach works. We're still actually trying to figure out how the brain works. It's the last part of the body. We don't really understand how it works yet. So this is why neuroscience is so fun and exciting these days. It's a big challenge to figure out how the brain works. So here's what we're trying to explain, and I'm just going to spend a couple minutes sort of going through leading up to Cajal and why what he did was so really, really important. The next person I want to show you is this guy, Felix Vic Desire, who lived during the French Revolution, basically. And he published his great book in 1786 that illustrated in beautiful detail, as a matter of fact, as well as has ever been done, the human brain. And this is one of the drawings from his book. He was another really, he was the greatest anatomist of his age, the 18th century. And he was a physician. He was Marie Antoinette's physician, actually. And she was, guillotined in 1793. And he died in 1794. People think of stress, basically. He was about 40, I think, at the time. But he was... So here's a guy, you'd think he's 80 years old, but, back in the day, this is what 35-year-olds look like. But here is one of his paintings, almost, really, of the human brain. Before the lecture, before you came in, I was, giving an anatomy lesson on all of this. But you can actually, they discovered by this point, actually, all of the parts of the brain that you can see in a modern MRI, for example, when they do brain scans. All of that stuff has been known for a couple hundred years. This is a magnificent atlas that uses color lithography, which was just invented like 5 years before this. It's got line and stipple engravings. It's hand-colored. I mean, these are phenomenal works of art, actually. And so by this time, by, let's say, 1800, they actually knew all of the basic parts of the nervous system. As well as what you learn in medical school now, really, the gross anatomy. This is like the brain is an organ, basically. But what then came along was the next giant step, and this guy Purkinje, you remember that name, because it's a weird name, but he was a Czech physician, scientist, and people, when you study biology, still you learn this guy's name. And what he did was discovered the nerve cell. So instead of looking at the brain as an organ, they finally got microscopes and methods that were good enough to slice the brain up really thin, put it on glass, stick it under a microscope, and what is the actual structure of the brain? And this was just about the time when people realized that animals and plants are made up of cells. And so he discovered nerve cells, which, we call neurons these days. But he went and looked at a lot of different parts of the brain, including the human brain, and saw that each place you look in the brain, the nerve cells look different. And so there's actually different structures in different parts of the brain. That's why you can see them. That's why they look different. They're made up of different kinds of nerve cells. And there's one part of the brain in particular that he became famous for. It's called the cerebellum. It means the little brain. It's on the back of your brain stem back here. And it's a simple layered structure. And what he saw in there, actually, back in 1838, was that this part of the brain actually had 4 layers to it. No reason to go through all the details, but 4 layers. And a middle layer here was made up of these really weird-looking sort of flask-shaped cells, big, giant nerve cells. And they're little extensions that come out of the nerve cell. And he saw going up going up in this direction, he saw some big pulled out extensions of the cell. And coming out of the other end was a tiny little string. So, it's an observation. And what does it mean? Well, at that point, who knew? But people knew this was a system, and they knew that nerve cells, or they figured nerve cells must be connected to each other to form a network, to form a system. How do those connections between nerve cells actually take place? How do you form circuits in the brain? That was the big question, completely unanswered in 1838. But you had all of these funny-looking different cells over here. This is a part of the brain. It's called the black substance. You can see these granules in the cell over here. This turns out, 200 years later, to be a part of the brain that degenerates in Parkinson's disease, substantia nigra. And so, all of this, that's the thing about neuroscience in the brain is localization. Every part of the brain has its own little function, and different diseases come out of problems in different parts of the brain. So Purkinje discovers in the cerebellum these giant flask-shaped cells. And, okay, what's the next step? Well, the next step was this guy, Camillo Golgi, who was a poor, small-time physician in a rural part of Italy, working in an insane asylum as a physician. And he had a lab in his kitchen, some chemicals in there, and he'd fiddle around and throw pieces of brain tissue into different combinations of chemicals. And apparently, purely by accident, one day, he'd left them in the chemicals too long, and he saw this black, stuff in the bottom of the jar, and I'll cut it up and look at it. And it turned out to be the most important discovery, actually, practically, in neuroscience, because it became known as the Golgi Stain, 1773. And remember those little Purkinje cells, the little flasks? This is what they look like. They're like trees. All Purkinje saw, here's one, here's two, and here's the third one. All Purkinje could see was a row of these things with the cut-off extensions coming out the top and a string coming out the bottom. Well, here's the cell, and these little cut-off things form these huge, huge branched complexes like a tree. It's on the cover of the book of The Beautiful Brain. And so these are a very characteristic part of a nerve cell. And look, he colored in red that little thread here that comes out the bottom of each one of these. And so nowadays we call these actually the three parts of any nerve cell. One is this tree up on top called a dendrite, and the string down here coming out, and every nerve cell has one. It's called the axon. And Golgi looked at this and said, God, these axons are coming out, and they're all coming down in here, and they form a net in there, almost like a fishing net. It's called a reticulum. So he thought the way that these dendrites sticking up were like the roots of a tree that were getting nutrients from the blood vessels. And then the axons were the sort of the wires in the circuit diagram. And he thought that all the wires came together and intersected and formed some kind of a net, which was sort of a current theory at the time. So I don't want to bore you with a huge amount of details, but this is from a physiology textbook in English from 1891. And here are some nerve cells hooked up in this sort of fishing net arrangement here, where all of these axons, these extensions, just go between the nerve cells, like this, forming a reticulum. And down here is the skin, and here's a muscle over here. So the idea is, let's say you pinch the skin, and a signal goes up here to this nerve cell, and then out these extensions to other nerve cells and down here to a muscle. So like when you pinch the skin and you withdraw like this, you step on something and raise your foot. The idea is that this signal would come up this extension here, go through the nerve cell, and just go out to the muscle to cause a reflex contraction. And that information would then go to another nerve cell over here, and another one might go out here, and you might actually get a contraction of 2 muscle cells out of here. So this was the theory, Golgi's theory, about how circuits in the brain are actually formed. It's called the Reticular Theory. All right, so now we come to our hero, Ramon Cajal, who was born in this tiny, the village today only has 200 people in it, I think. I mean, it was this really poor village in Aragon, up in the Pyrenees near the French border. So he couldn't have been born into a more remote area, and he's essentially completely a self-made person, actually pretty much like Golgi. His dad, by the way, though, was the local doctor. He's almost a self-taught guy, and in a tiny little village here, being the doctor was sort of important, but he was no great physician or anything. But he was born in 1852, and he, as a kid, he was really mischievous. He hated authority. He wrote a great autobiography. Actually, it's one of the 2 great autobiographies in science. The other one's Darwin's autobiography, but Cajal's autobiography is great. It's wonderful reading. And he describes himself as a mischievous little kid. He'd love to go out with his buddies and throw rocks at little old ladies and, do all kinds of stuff, get in trouble. He didn't like school particularly, but he loved to draw. He called it graphomania, basically, but he would buy notebooks and just draw everything he could. He filled up notebooks. And his father thought this was a trivial pursuit for his son. His son should become a doctor, and did everything he could to dissuade him from doing it. But, Cajal kept it up and went to some little art school nearby and, started to learn how to draw figures and still lifes and that kind of stuff. But when he was 16, he read a book on photography, and he built his own camera, made his own negatives, made his own paper, took pictures, developed them, all of this. And this is when he started taking these self-portraits. This is the first self-portrait. When he was 16, he took of himself. He had just read Robinson Crusoe, in school. And so he decided to go out back behind his house, and pretend like he was out in, somewhere, and made the bow and, posed in this loincloth, But I showed you that self-portrait he took after he won the Nobel Prize. And he's a little bit like Rembrandt in that he took a huge series of great self-portraits throughout his life. I mean, here's one. He went through a muscle building phase when he was 18. So he was incredibly compulsive, actually. If he wasn't drawing something, he was muscle building. He took up chess and got to the point where he played 20 people simultaneously. He just did all of this stuff. And as life went on, here he is, Hamlet, contemplating, finally his dad got him to go to medical school, right? And so here he is as a medical student, basically, contemplating a skeleton. But, he went on, and he actually had a family. He had a, loving wife, and I think he ended up having 6 kids. Here's a self-portrait with 4 of his very handsome kids, I must say. And here he is. This is this really famous self-portrait with his microscopes and all those chemicals in the background that you'd throw tissue into. Histology is the name of the science of studying tissues under the microscope. And, just a absolutely wonderful self-portrait. And he was about 34. He was in his early 30s. And he had a position at a medical school teaching histology, basically. And he decided to write the first book in Spanish on histology for medical students in Spain. And so he was illustrating it himself, and he was really thinking about this. And he said in his autobiography that he realized the biggest problem in the 19th century in understanding the nervous system was how do nerve cells talk to each other in circuits in the adult brain? What is the mechanism for forming circuits in the brain? Is it this reticulum that Golgi thought it was, or what is it? And he didn't buy that, but he wasn't sure, but he thought that was the great unsolved problem. So he went, a friend of his had been to Paris, which was a leading center of science at the time. You know, Spain was, let's face it, fairly third world in the 19th century. So they would go to Paris to learn the latest methods, and his friend came back, and Cajal went over to his house, and they sat around and looked through the microscope. And one of the slides this guy showed Cajal was of the Golgi method. And Cajal said, he was 34, he looked down the microscope and realized this was going to answer that question. How do nerve cells talk to each other in the adult brain? And that was 1887. 19 years later, he won the Nobel Prize. And he said he worked 16 hours a day, 7 days a week, trying to figure this out with sleepless nights every time he discovered something. So, you know, this compulsive behavior of his, combined with being a brilliant artist, is what actually made him. So here's this drawing of Golgi's, and these are the famous Purkinje cells over here that we talked about, and here's this so-called reticulum. So Cajal spent about a year just making the Golgi method better, and the first place he looked was in the cerebellum, this place where Purkinje cells are, of birds, actually, young birds, where you could really see things. And his first discovery was this, and takes a little bit, you know, it's a beautiful drawing, but see this row of lightly shaded cells over here? Those are Purkinje cells, actually just like Purkinje drew them. They're unstained in this Golgi preparation, unstained, so there are little shadows there. But what he saw was a nerve cell right here. Here's the cell body, and here are all these dendrites sticking out, the bushes sticking out of the top. And here's the one axon, the one thread coming out of the cell, and coming along here on top of the Purkinje cells. And for each Purkinje cell, there's a little branch that comes down and forms a basket around each Purkinje cell, almost like ivy growing over them. But you'll notice the Purkinje cells aren't stained. This basket cell is individual. These cells are not connected directly, okay? And he saw here, this is not a reticulum. These are individual, separate cells. So he called this a basket cell, and it's a really famous cell. You see these baskets around here. And he then went on in the next 15, 20 years, actually 30 years, to look at every single part of the nervous system. And this is a huge job, huge job. And he looked in different animals. I mean, he looked in fish, and he looked in frogs, he looked in human, he looked in mice, at all parts of the nervous system, basically, and saw the same thing. Everywhere you looked, you saw individual nerve cells. They didn't form a net, a continuous net. And this is one of the most famous ones. This is in your cerebral cortex, the part of the brain you think with. And here's the cell body, and it's got, see the dendrite? There's one dendrite that sticks straight up here, a big thick one, and then a lot of other little dendrites coming off. And then here's one extension here, the axon, this thread that comes out and branches over here. And you can also see on these dendrites little spines, tiny little spines coming off of there, which Golgi thought were artifacts, and Cajal said, no, they're absolutely important. And so what Cajal did with all of this thinking was he didn't just draw these, and every part of the brain he looked at had a different shape. This is called a pyramidal cell because the cell body is sort of pyramid shaped here. okay, with the top of the pyramid going up like this, versus a basket cell versus something else. And so what Cajal actually did then in the next 15 years was to take this gross anatomy of Vesalius and 350 years later describe what's going on in the circuitry at the level of nerve cells, okay, of individual nerve cells. The individual nerve cells are like Legos. They're individual things that you piece together. And with Legos, you can build a little cube or you can build a cathedral, right? It just depends on how you put those Legos together. And so the nervous system of a human is put together in a certain way with nerve cells, and the nerve cells of a worm are put together in a different way. But here's the gross anatomy, and here is Cajal's explanation of what's going on at the cellular level. So here's the brain up here, and here's the cortex. That's this little part of it here. Over here is skin, and over here is muscle. So it's a little bit like that really schematic thing I showed you before. But instead of information just going into a nerve cell and coming back out to a muscle, no. Here's the skin, and if you poke the skin here, you send information up through this axon, this cell body here, into the spinal cord over here. And so this is how sensory information gets in through this sensory nerve cell. And over here in this spinal cord, here's another nerve cell that has an axon that goes out and innervates, goes to muscle cells. And up here in the cortex, here's a pyramidal cell. Here's this pyramid-shaped cell body with the top of it going up. And here's this axon coming out and going all the way down to the spinal cord and ending over here next to a nerve cell. So if you think about moving your foot, for example, lifting your foot, the signal comes from this nerve cell in the cortex, goes down this axon, and goes onto the dendrites of this cell here. And its axon then goes out to the muscle to contract it. So he actually explained reflex control of your muscles and voluntary thinking about it based on this cellular organization. So number one, the elements of a circuit are individual nerve cells. And the other brilliant thing he came up with, brilliant idea, hypothesis, was that nerve cells, this funny shape of dendrites sticking out at one end and an axon coming out the other, he said that information always goes into a nerve cell through the dendrites and out to another cell through the axon. So actually nerve cells, information in a circuit only goes in one direction. It doesn't go everywhere. And so he overthrew this whole idea of Golgi's, that there is a reticulum or a net. Instead, there are individual parts of the circuit that are linked together in very specific ways. And one reason this was so important is that this axon comes up and ends on the dendrites of this cell up here, and then the axon comes down. But when you have the interaction between this axon and this dendrite happens at a very specific little place called an axon terminal. And it was very soon called a synapse. So there's a very localized place where nerve cells talk to each other, or they talk to muscle, called a synapse. And why is this important? Well, Cajal theorized back in the 19th century that maybe this little synapse, this interaction between two nerve cells, is where learning takes place. You know, it's like when he was a muscle builder and you exercise and your muscles get bigger. Maybe if you use a certain part of a circuit, it strengthens. And that strengthening could take place at the junction between two nerve cells. So he hypothesized that learning could take place when you have a gap or a relationship between two neurons. And it took another 100 years pretty much to prove that, but that turned out to be absolutely right. So Cajal revolutionized the way we think about the organization of the nervous system. And if you take biology in high school, college, graduate school, the way you learn about neuroscience is this, basically this model, this Cajal model of the neuron. So this, he finally got enough evidence together by 1899, 1904, this period here, to write this 2-volume book of research on, it's called the Histology of the Nervous System in man and vertebrates. 887 original pen and ink drawings, which you'll see a lot of them out there in the gallery, not a lot. There's only 80 out there, but they're probably the best ones. But 1,775 printed pages. It's all his own research. I mean, there's essentially no review of what people thought. This is what he thought. It's a brilliant, brilliant book. And so, 1906, they gave the Nobel Prize to both of these guys, Golgi and Cajal. They had absolutely opposite theories about how the nervous system is organized. One of them had to be wrong and one had to be right. And they weren't entirely sure in 1906 who was right or wrong. It's actually a great story. They probably gave it to Golgi because he invented that method. It never would have gotten done without his method. But having said that, he also did a lot of beautiful work on the nervous system. So it's a great story. And here are these two guys. By the way, if you remember, Golgi always had that mustache. It just keeps getting better and better. But, you know, there was huge animosity. This is a great cartoon. It was done a few years later. But Golgi really, really disliked Cajal because he realized Cajal had probably, you know, outsmarted him. But, so, you know, this is after the Nobel Prize, basically. And here's the famous Golgi mustache and Cajal's beard. But, you know, here's Cajal's knife in the back, you know. So science is not always, you know, real pure and everything. And, you know, by this time, Cajal was just really a famous character in Spain, actually. Here's a great caricature of the medical faculty at San Carlos. And what you can see, you know, here's, I don't know, here's the pediatrician over here, and, you know, here's a clinician getting a rabbit out of the bag. But these are the different doctors. And Cajal wanders in over here, and he's drawing one of these pyramidal neurons in the back of this guy over here that has no idea what's going on. Yeah, right. So there's sort of a reason I picked some of these drawings to show you, you know, because there are these themes that run through there. But he was, you know, Cajal was really famous. And you know, then it was, so he described in this monumental 2-volume thing that Neely and I translated, the organization of the nervous system in vertebrates. But then he went on and wrote this 2-volume book of original research on what happens to the nervous system when it's damaged or during disease. So if you have a stroke or you cut a nerve or you have some damage, you have a tumor, you have something, how does the brain try and regrow, or the nerves regrow? What are the mechanisms underlying the regeneration and the degeneration of the nervous system? So what happens when you cut a nerve, actually? You know, look at it scientifically. What happens to the axon and the cell, and what happens in the rest of the nervous system? So this was a very, very important. So he actually wrote 2 great books, and many more. I mean, this guy was incredible. And so by this time, I mean, he was really famous. And he set this one up, too. Here he is giving a gross anatomy demonstration to some of the faculty colleagues, and he's got a scalpel over here. When I was putting this together today, I was wondering, I wonder if that's one of the colleagues that just laid down on the table there. That guy doesn't really look that dead to me, but I'm not quite sure. But, you know, let's face it, Cajal was an egomaniac because, you know, this is this fabulous, you know, painting of Dr. Tulp's anatomy lesson by Rembrandt in 1638, right? So, I mean, he really just set it up to copy that, you know. I mean, right down to, you know, Dr. Tulp here is holding up a tendon with this, and, you know, here's Cajal over here with a scalpel in his hand. I mean, you know, it's just great. It's absolutely great. Really interesting guy. And, you know, here's a monument to Cajal, just to show you how famous he is in Spain. This is in one of the great parks in Madrid. This was erected while he was still alive. He was, he had another 6 or 7 years to go. So, I mean, it's pretty amazing. And when he died, there were thousands of people. This is the funeral march down one of the main streets in Madrid. And here's his coffin over here. And people, all kinds of, they say people from every walk of life came to honor him. When you look in here, I mean, it's interesting. Here's, you can, I saw some kids in here, and it's amazing. He was just exceptionally revered. I'm not quite sure what's happened in the last couple of years. That's a good thing to talk about. But, actually the last book he wrote, this was published the year before he died. He died in 1934. He still had to write a book called Neuron Theory or Reticular Theory in 1934, justifying his claims over Golgi, basically. It still wasn't, there were still a few people that held out. This is a English translation that was published a little bit later. But, he was so passionate that at age 83, he felt he had to write a book justifying his scientific career, basically. So it wasn't actually until the 1950s when they absolutely proved that Cajal was right and Golgi was wrong, that there's a gap between two nerve cells, or between a nerve cell and a muscle cell. And it took the electron microscope and a magnification of like 60,000 times to see this. Technologically, it took an incredible breakthrough. So when you look at a slice of brain, a pyramidal cell, let's say in the cortex, under the electron microscope, this is what it looks like. Very different than a Golgi impregnated neuron or anything else. But here's a dendrite over here. So remember, this is the receptive part of a nerve cell. And this funny looking thing over here is an axon terminal. So this is the end of an axon. There's a little swelling there that ends on a dendrite, and there's a very thin, very incredibly thin gap between those two. You could never see it even at the light microscopic level, actually. And so this proved, this region right in here that's sort of dark is actually where the transfer of information takes place between two nerve cells. And when they got the electron microscope, they could see in this axon terminal, see all those hundreds of little circles in there? Those are little balls, they're little vesicles, they're little packages of chemicals called neurotransmitters. And the neurotransmitters, the molecules in these little bags here come up over here, and when information comes down the axon, it triggers the release of chemicals from these little bags that act on the dendrite to transfer the information over. So by the 1950s, they started to realize that the way nerve circuits work is electrical signals going up and down the axon and dendrite, and then between two nerve cells, there's a chemical transfer of information. So neurotransmitters are really, everybody knows now about neurotransmitters, and one reason they're so important is because the pharmacology, all drugs that act on the nervous system, on the brain, basically act on neurotransmitters. They block neurotransmitters, antidepressants and anxiety, all of these drugs act on neurotransmitters in the brain. So this is, the electron microscope led to this whole revolution, the next revolution, really, in understanding how circuits actually work, like the chemistry of circuits. And, just to bring this full circle, I wrote an article actually just a couple months ago, last year, called From Cajal to Connectome and Beyond. And the idea, so Cajal is still incredibly important in people's thinking about the nervous system. And there's this new word that you'll probably start running across in the newspaper in your reading called connectome. And it is it derives basically from the genome. And this was this giant push in the basically 60s for 30 years or so to sequence the human genes, to figure out exactly what the structure of genes are, which has led to personalized medicine, basically. But this was all based, actually, on another pair of young guys at Cambridge in the 1950s, 1953, actually. This is Francis Crick and Jim Watson over here. And they were students. Crick was working on his PhD, and Watson was a postdoc, I think from Indiana, University of Indiana, that went over there. And so on their spare time, at 5 o'clock every day, they'd have cocktail hour. Was it the Golden Eagle? I forget. There was some pub there in Cambridge that they went to. And started thinking, what could we do to get famous? We're sort of working on our PhD and this and that, but honest to God, they said, what could we do? What is the most important question in biology? It was sort of like Cajal thinking, what's the most important thing I could do? And they said, well, heredity. Heredity. What is the basis of heredity in all living things? Plants, animals, amoebas. Well, it's in the genes. It's in the chromosomes. So they decided, why don't we figure out the structure of genes, of chromosomes, because they're involved in heredity. And so they actually just did a bunch of reading and got some tinker toys and put all the data together, basically, and constructed a model, a physical model, based on all this data about what the structure of genes, of chromosomes could be. And they came up with this double helix model that they published in 1953, which led to all of molecular medicine today, the understanding of genes, because it turned out to be right. And so this stresses really the importance of having a really good physical model of something you're trying to explain, which is what Cajal really did, was start to create a physical model of the brain that we could understand, begin to understand how it works. So here's this wonderful book that came out of the exhibit that you can either see before or after or some other point today. The Beautiful Brain, these drawings. And this is a great book, but what you really have the treat of going in and seeing is the original drawings that he actually did. And you don't really have to understand the science all that much to really appreciate how beautiful actually these drawings are. But I think it helps to actually, like anything else, to know a little bit about the background. And one of the great things, I was here for the opening at the end of January, and they probably still do, but they have magnifying glasses in there on the wall. And it really, go over and get one of those and go up to one of these drawings and actually look. These are pen and ink. He did a pencil outline and then he inked them in pen. And some of these lines are so fine. He must have taken individual bristles or something. It's incredible to look at the detail in these things. So thank you very much for coming tonight, and enjoy the exhibit.
Students with microphones, okay? Anybody have a question? Yes. Great.
Thank you for this wonderful talk.
Thank you.
There must be some control that is probably genetically determined as to how we learn, how we develop emotions. All kids learn to learn more or less the same way. Certain experiences cause sadness or happiness. There's some patterns of response from the brain to learning, to emotions, and this must be regulated genetically because we all share that. How much is known about that?
Well, yeah, so the question is, the emotional side of our consciousness, what about, is there a genetic component to that, or how does that work exactly? And actually, that's the sort of the thrust of my own research, but almost all of this thinking goes back to a book that Darwin wrote, actually in the 1860s, called Emotions. I forget now, Emotions in Men and Animals or something like that. But he postulated that emotions are a product of evolution. And they are, for example, just think about a baby that is hungry. starts crying because it's hungry, and mom is right there and feeds. And so there are the basic emotions are what actually keep us alive. If you're faced with a big threat, let's say, think about animals that are faced with a predator out in the wild, for example. It produces a feeling of fear, which motivates that, which mobilizes that animal actually to defend itself. And so there are these basic emotions, actually, that are shared by animals and even people, that are hardwired into the brain. And when you look at, these are the circuits, actually, that we're trying to figure out based on how Cajal said, think about circuits. And so just like the control of muscle, and you could see in Cajal's drawing where you could get a movement based on a reflex. You know, if you poke your skin and you move away, that's a reflex, but you can also think about moving it. So there are ways of controlling the emotions. There are actually emotions that are just triggered by things you see, they're reflexes. But then you have this cortex up here that you think with and you learn with that modifies the instinctive behaviors. And so like in people, even, I mean, if you have pets, if you have dogs, cats, you realize that they learn how to control their emotions to some extent. So yes, they're hardwired, and they're basically, I mean, people talk about these as, the basic ones as being basically instinctive. In other words, they are part of your genetic makeup. And then you learn how to control them, and you actually learn how to modify them. You learn how to do things better. And what we do is we learn how to avoid those problems if we can. So, instead of being hungry every 3 days, we learn how to eat 3 meals a day and avoid that. So there's a lot of learning going on. But the idea is then that there's a genetically hardwired instinct that keeps you alive. And then there's learning on top of that that actually makes the responses better.
Hi, thanks for the talk. It was awesome. Is there any known understanding about how new dendritic connections are formed? So if an neuron upstream is sending a 1 or a 0 in an axon, how does a downstream neuron want to go connect with that one and not with something else? And
yeah, so it gets at how does so the brain at some level, the buzzword is plastic. In other words, it changes based on experience, and from the moment you're born, really. And so people have wondered how, and you can actually see these changes under the microscope, but people ask, how does it happen? And they want to know now what's the chemistry of it, basically. And, part of this, in all honesty, people are working on drugs that'll make you smarter, that'll help synapses form faster, for example. Or people with post-traumatic stress syndrome, can you make certain memories go away? Traumatic memories. So they're trying to figure out exactly what these mechanisms are. And most people think it boils down to neurotransmitters and the receptors for neurotransmitters. So neurotransmitters are released from the axon and act on the dendrite. So it's a chemical going across to the dendrite. And there has to be a molecule on the dendrite that recognizes the neurotransmitter. So what people have found is that some neurotransmitters come across, and under the right condition, they open up channels in the membrane. More calcium goes in, it triggers a stronger synapse, basically. So they're getting to the point where they actually can understand the chemistry of what's going on when a synapse gets strong, when you learn, basically. There's a really famous guy named Eric Kandel at Columbia who won the first Nobel Prize in understanding the chemistry of memory, learning and memory. Yes?
What makes those little sacs start? What starts?
This is, yeah.
And the snaps, is it the electricity? Is the id in there? We haven't seen it yet? What's the
Well, there's a unique thing going on here at the Weisman. Actually, you can go out and see an exhibit by Cajal, by Freud, and by Darwin out there. And Darwin and Freud and Cajal were contemporaries and actually never met. And the interesting thing is Freud was a neurologist, and he spent the first 10 years of his medical career in Vienna as a neuroanatomist, actually. He invented a method for staining nerve cells. And it got to the point after about 10 years when he said, unlike Cajal, he said, I am never going to figure this out in my lifetime. It is too complicated. He realized. And he said, instead of studying the brain, I'm going to study the mind. And think about the mind in the same way you think about the brain. What are the parts and how do they interact? So he came up with these basic parts, ego, id, superego. And what are the connections between those? What's the dynamics between those? So ever since, people have been trying to figure out, is there a mapping between Freud's thinking and neuroscience, between the brain and the parts of the mind? And, there's a lot of theories, but nothing really holds. So at one level, it's all chemistry and electricity. On the other hand, people really want to know, how does this actually get set up in the first place? In the baby, in the embryo. How do these things actually form to begin with? Or new learning. Well, new learning is chemistry, basically. Well, okay, so if you push me on this, and there was a really famous guy named Donald Hebb. He was a Canadian neuroscientist back in the 1940s, psychologist actually, who was the first person to think about like Cajal's ideas and learning and memory. And he said, maybe if you have a nerve cell that goes somewhere and does something, the axon goes somewhere, and you have two inputs, two nerve cells coming in. And normally their information is coming in at different times and it's sort of random. But if information actually came in at exactly the same time from two sources, maybe that would trigger a strengthening. So something out of the noise. And so people have actually found now that when two signals come in at exactly the same time, it opens up more channels. You get a stronger response, and that stronger response lasts longer. It's called long-term potentiation. So they've gotten to the chemistry of this, and a lot of it has to do with timing. It's almost all timing. If two things, and this is, we're sort of getting out in left field here, but there's something called associative learning, and that's when two things happen at the same time. You remember them. Pavlov's whole thing was associative learning. He put a plate of food. Actually, he showed a dog a piece of meat, and the dog started salivating before he ate the meat. When you chew and you eat, you secrete saliva. It's a reflex. But dogs, when they see it, it's an anticipatory thing. Well, what Pavlov did, he said, what if I rang a bell instead of showed him a piece of meat? Well, guess what? Nothing happened. But if he rang the bell just before he showed him the meat, the next time he just rang the bell and the dog started salivating. It learned an association between the food and the bell. Something that happened at basically the same time. One was food, one was a bell, but if it happened at the same time, you could take the food away and the bell would work. So the timing is really important. It's really important because that triggers a bigger chemical response, basically. And as a matter of fact, not just a bigger one, but almost a different one.
Yeah, you've got the microphone. Go ahead.
A couple of questions. One, when I think of chemistry, I think of the chemicals getting used up. So if you maybe touch on that, the idea, and then the other one is, I don't know, do you call it muscle memory? If I touch a hot stove, I pull back before the signal gets to my brain.
Yeah.
My muscles learn to pull back. So if you comment on that, how did Cajal figure that little nuance?
Yeah, he really did. And the first question again was?
The chemicals getting used up.
Yeah, so, here's the thing, when
in that transmission process.
Yeah, so, right. So, dendrites, cell body, axon. And what people realized was that when you cut an axon here, this part of it away, this part farther away down here dies, but this part up here remains. And what people began to realize shortly thereafter, all of this stuff is synthesized in the cell body. The manufacture of chemicals, by and large, takes place in the cell body here. And these materials actually continuously ship down the axon to the terminal. And so when you disconnect here, so basically the factories for making the chemicals are in the cell up here. And they're constantly making them, and they get used up down here. And well, it gets a little more complicated because there's actually recycling down here. So they get released, and then they get taken back up and recycled. So there's a recycling mechanism. And then there's a constant replenishment.
There, use this. There you go.
And then there's a constant replenishment. So actually, we're sort of getting into the whole cell biology here of the nervous system. But this is how, after Cajal, neurologists could actually look at symptoms in neurological patients and figure out where the problem in the central nervous system was by knowing what the circuitry was. Because if you lost sensation in one little part of your body over here, it meant that the particular nerve that went to that part of the skin, they know exactly what nerve that is and where it goes into the spinal cord, and they actually know what part of the cortex you feel that with. Now, muscle memory. So this diagram also explains that, because here's the muscle over here, and here's a stimulus to move that muscle. Let's say you pinch the skin here and you move this muscle. This all takes place in the spinal cord. So if you have a paraplegic or a quadriplegic and you pinch the skin, they'll move their foot, the muscle. And they don't feel it. They actually don't feel it, because that sensory information not only goes to the muscles, it goes up into the brain, up into the cortex, where you actually feel it. And so you actually move the muscle before you feel it, because this reflex down here is a shorter period of time. It takes time for that information to go up here. And so Cajal explained this difference actually between reflexes and voluntary control of the same behavior. This is very profound stuff. This one diagram here that was drawn in 1893 is absolutely brilliant. I could talk about this one slide actually for the hours. But so this is based on all of this. People that have absolutely intractable pain from cancer or certain things, where actually drugs, even opiates, don't do anything. The last resort is to come in here and cut this sensory nerve over here so it doesn't get into the central nervous system. But depending on where the pain comes from, a surgeon knows exactly which nerve to cut, because they've got a blueprint for it. They've got a wiring diagram, which pretty much all started with Cajal. If you've got paralysis in a certain muscle, it's because the nerve to that particular muscle is damaged. And you don't even have to look into the body. If that muscle is not contracting, you know which nerve it is. You know where it's coming from in the spinal cord. So a really good neurologist can figure out a huge amount from your symptoms where in the nervous system the damage actually is. Nowadays they go in and do an MRI, let's say, and they can see a lot, but they can't. It doesn't have a whole lot of resolution. So Cajal actually revolutionized neurology. He absolutely revolutionized neurology.
Another question. Any more questions over here? Yeah. Why don't you take that?
Oh, here. Yeah.
Back there.
Hi.
I wonder if you could explain how, especially in the central nervous system, Cajal inferred the direction of information flow from structure, and then also, before we understood axonal currents and chemical synapses, what did he think the information was? What form did he think it took?
That's great. That's a great question. See, he was one of the few people, he drew arrows on his anatomical diagrams, coming in, going down, going up. This was pure speculation based on thinking about what things probably happen. And the way he really did this was, if you have a muscle, you know that the spinal cord is controlling that muscle because you can cut a nerve and it doesn't work. So he knew that information from the spinal cord was going out the spinal cord to a muscle. So he could actually say, well, I know what direction things are going in this one. And when you pinch the skin and stuff's coming in, he knew it had to be going in this direction. So then he looked in the eye, and he looked at the way the nerve cells look in the retina, and you could see exactly the same thing, dendrite to axon, dendrite to axon. And he did that in every sensory system. He did it in the motor system, and then he guessed that the same was happening for any neuron with dendrites and an axon. So you could look in the cortex and say that's got to be the input side, and that's the output side. So it was all speculation. It's brilliant speculation, and it wasn't really until the 1930s and 40s and 50s when they could actually get at the physiology, the actual physiology and chemistry of all of this. So it took a long time, but it was, they had a hypothesis. They had this is a scientific hypothesis about how it works. He didn't know it worked that way, but that was the best guess, and it turned out to be absolutely right. Yeah.
Professor Swanson, I find it interesting you're characterizing this as speculation. When they had 100 years prior to this known that the electrical impulses were going to travel along axons and so forth, so they knew about that. Did that enter into what he was thinking?
Yes. Well, I say it's speculation because this is pure anatomy. Right. It's really, and it's just looking at dead tissue. And so, actually, to be fair about this, in 200 AD, Galen clearly saw that there were sensory nerves and motor nerves. For example, the optic nerve from the eye going into the brain is taking the visual world and taking it into the brain. That's a sensory. Then there's a nerve that moves your tongue, for example. That's all it does. And if that nerve is cut, your tongue is paralyzed. And they actually knew that at that point. And so they knew there were, but what Cajal really did was this idea that the dendrites were the input side, the axon was the output side. And he said it has to be true in the retina. It's, but when you get into places like the cerebellum or, these places in the brain, it's speculation, but I must say it's a high-grade, very high-grade, a hypothesis. But I think just to be strictly true, it's guesswork. But when you have a genius and you have somebody that's really not just making little drawings, when you go in and look in the exhibit, every single drawing in there tells a story. It's not just a photograph of something under the microscope. He thought about, and every single drawing in there tells you something. You can actually use it to teach, because it has there's more to it, which I think, that's probably one hallmark of, really great art, is that there's a lot of added value to it. Yeah.
Obviously, Cajal was a revolutionary genius, but was he wrong about anything?
Yeah. He actually said no matter how great any scientist is, they're minuscule in comparison to what you actually want to know. There was some stuff we were talking about just now. There's a concept called inhibition. And when you get into really complicated circuits, you say, well, this nerve cell goes to that cell, activates that cell, and it goes to the next cell. Well, if everything activated everything, you know what that's called? It's called epilepsy, right? Because it's totally out of control, right? Everything's excited. So it turned out in the 1950s that about half of the nerve cells in the brain inhibit instead of excite. So there's a balance in there. And when that balance goes out of whack, you get something like epilepsy. And so this idea that circuits actually, that a nerve cell can actually stop the next nerve cell and that you have these combinations in there of plus and minus, stop and go, escaped Cajal as it escaped everybody else. That's one of the biggest things he was wrong about. But he covered himself. He was a biologist, and he said, these are sort of rules, but he said, wouldn't surprise me at all to have exceptions to these rules. And it turns out there are exceptions to all of these rules. There are little places, actually, where two nerve cells actually are fused together. But this way of thinking about circuits is sort of the starting point, at least, for everything. So find other stuff that he was really wrong about. When it came down to his absolute microscope observations, he didn't make too many mistakes. When he started speculating, then things started to go haywire. There's just a book that came out last year. When you talk about Freud, Cajal hated Freud intellectually. And so, he said that this can't be right, that there's actually a structure to dreams, that they mean something, and, profound about your personality. So when he was I forget now, but he was in his 60s or 70s, he started keeping a dream diary. And so he started recording his dreams, and somebody just found the manuscript and translated it and published it. So you can find a book on Cajal's dreams and what he thought about it, and he said Freud is just full of it, and there's no rhyme or reason to any of this, but his dreams are full of stuff about his father, who hated him, being an artist and all of this stuff. So actually, it's a case study actually in Freudian psychology. He also wrote a book of science fiction short stories. I mean, he's phenomenal, and they've been translated. He's still in print in Spain. He wrote a book they're called sort of cafe aphorisms, famous Spanish sayings people use around when they're drinking and talking in Spain. And it's really popular, and it's still in print. You can buy a copy, people still. He wrote a book called The View of a World, a view of the world of an octogenarian, an atherosclerotic octogenarian. He was starting to develop, a little bit of dementia when he was 80, and wrote a book about his cognitive decline and what the world looked like to him when he was 80. I told you his autobiography is brilliant. This guy, he wrote the first book on color photography, the theory of color photography. This guy is amazing, and I think he's always been like an icon to neuroscientists and stuff. But I think with this exhibit and more and more coming out about him, he's a fascinating character. They actually did a miniseries, a soap opera on him in Spain. Like an 8-part miniseries. There's a lot of stuff about him, personal stuff that's interesting. Interesting guy.
Thank you, Larry. I will just say I've read his autobiography, and it is fascinating, even for an art historian. And I have this idea that he actually learned by drawing, and that a lot of his ideas came out of the fact that he drew. So for all of you scientists here, get out your pencils and draw. Thank you very much. Thanks all for being here.